All right, hello and welcome everybody to our monthly virtual roundtable hosted by the Dairy Grazing Alliance. My name is Jessica Matthews. I am the apprenticeship manager. I'm sorry that my camera is off. It's better for bandwidth if it's that way, but I am here and I am listening. I'm going to turn it over to Lane shortly. Just as a little bit of housekeeping, if you have any questions, you can put them in the chat and I will grab those questions and ask Lane for you. And if I'll also put my information in the chat, so if there's any follow-up needed, you can reach out to me when we're finished. So with that, I'll let Lane take it away.
All right. Well, thank you, Jessica, and thank you all for having me. I'm excited to be talking with you guys today. So today what I'm going to talk about with you guys is genetics and basically the drivers behind genetic progress. But before we get into all that fun stuff, we get the obligatory introduction for who I am and who I work for. So, like Jessica said, my name's Lane Hischke. I grew up on a dairy here in Northeast Wisconsin. And over the past 4 years, I've worked in the genetic industry, doing everything from relief breeding all the way up to more of a consultant role, where I was designing and implementing genetic strategies for dairies of all sizes, really all the way across the U.S., which is very interesting just because of the variation of both the ability to implement complex or simple strategies, as well as just kind of the genetic goals for individual farms. So that's a little bit about me. And then the company I work for is Geno Norwegian Red. So for all 4 of those years that I've worked in the AI and genetics industry, Geno has kind of been there. I worked with a partner for a while, and now I'm directly employed by Geno. But basically, we started in 1935 in Norway. And at that time, it was very difficult, and there wasn't a lot of exchange of genetics within different valleys all through Norway. So for those of you who don't necessarily know, Norway is an extremely abrupt country, I guess. It's got all the fjords that cut through it. It's got valleys. It's got mountains. There's not really a lot of flat land. So each one of these valleys had kind of its own individual, I don't know if I'd go as far as to call them a breed, but definitely a genetic signature in each one of these valleys. And in 35, the idea was kind of came together from all these farmers to start a co-op to find the best genetics within all these valleys, share genetics, and make genetic progress. And a lot of the things that were important to these farmers were, of course, health and durability, but also ability to thrive on forage. So like I said, the weather and the topography in Norway is fairly extreme. There's very little corn grown in the entire country. There is some small grains like rye, but the majority of what the cows eat is going to be forage, just because that's what they have there. And with that, they also need to have high health and durability traits, because these cows are out on pasture every day. And in 35, it was difficult for a vet to get into the farm. And since then, things like antibiotic restrictions and expensive treatment has really made those health and durability traits even more valuable for Norwegian farmers. And obviously they have their place here. And then probably over the last 5 to 10 years, we've really made a lot of strides making the Norwegian Red productive in terms of U.S. farming. There's definitely a difference. So we've made strides there that have made them a competitive breed to use in crossbreeding, and kind of my go-to to breed Holsteins too anymore. So yeah, that's more or less about us. Now we'll get done with all that boring stuff and get on to what you're all here to learn about. What is genetic progress? So it's basically the improvement in desirable traits or the reduction in undesirable traits, according to one farmer. So they tend to work towards a genetic ideal. And what's important to know about that is the genetic ideal is kind of in the eye of the beholder, right? So for some of you, it might be that this red and white Holstein at World Dairy Expo that is the genetic ideal of a cow, and that's what every cow should look like. However, I would guess that a handful of you that are listening anyway probably like cows that look more like this middle group, more of a moderate frame, crossbred cow that's going to do well on grass, low input style cow. And then finally, the most extreme difference from that Holstein we were talking about earlier is this Angus mama. How did we get Angus and Holstein to look so different? Genetic selection and genetic ideal. Jessica, do we have some chats down there?
Nope, just so far so good.
Sorry, I got 2 notifications looking down there, so I won't worry.
Well, I just put yours and my email in there so folks can use them when we're finished.
Fair enough. Well, we'll drive on then. So as far as genetic progress goes, this is kind of the nuts and bolts of how it works. So I know that equations aren't necessarily everyone's cup of tea, so we'll try and break this down step by step. But this is essentially how we make genetic progress. So all of these 5 things are inputs. Selection intensity, accuracy, and genetic variation are all in the numerator, which I know, big words again. But basically what that means is we want this number to be as high as possible, and as this number increases, genetic gain will also increase. Next, we got this generation interval, and that is in the denominator and it has an indirect relationship on genetic gain. What that means is as generation interval increases, genetic gain is going to decrease, or we're going to get less genetic gain. And then finally, we're going to subtract off inbreeding depression. So the lower inbreeding depression, the more genetic gain we're going to get, essentially, if all of those work independently. Questions there? So now kind of jumping into the nuts and bolts of how this all kind of works. So now we're looking at selection intensity. And realistically, selection intensity is just the ratio of candidates that are allowed to contribute their genetic material to the next generation. So that basically means what animals are good enough and which ones don't make the cut. We've been really leveraging selection intensity very heavily since the 60s on the bull side, when AI came into being. But there's a lot of ways we can do it and a lot of management factors that kind of play in there. So as an example, the difference between bull breeding and AI breeding, that has a huge difference in selection intensity. So if a bull makes it through AI and is going to market with one of the major studs, it's 1% of 1%, which basically means that 99.99% of bulls born in the US do not make the cut. And that probably includes your neighbor's bull and jumper bull. Now, I'm not saying that AI is the only way to farm profitably, and I'm very aware that some farms don't have the facilities to breed AI for everything. However, the jump in genetic quality of frozen sires versus live sires is extreme and should not be undersold. If you really are serious about genetics and want to make genetic progress, sell every one of your breeding bulls. I hate to say it, but the next is kind of a less extreme example, but it's that ever classic question of sire quality versus sire price. Is it worth paying extra for those really elite sires, or are we better off maybe settling for an 80th percentile sire that doesn't cost me as much? And I don't have the answer for everyone for that. It definitely depends on the individual farm, as well as maybe repro statistics and some of those other things. But if you ever want to talk through it, my email's in that chat. Feel free to shoot me an email or give me a call. I'm happy to kind of walk through that on a more individualized basis. And then finally, the last place where I see selection intensity kind of get ignored in some cases is the number of sires used on a farm. Using a lot of sires isn't necessarily a bad thing. There's a lot of good reasons to do it. It spreads your risk, can give you some of those lines, and maybe you're doing some corrective mating stuff on a more individualized basis. However, my question to you is, how much worse is your 16th best sire versus your first best sire? And could we make more genetic progress by maybe only using 8 sires a year that are the best 8 rather than including those next 8 after that? So again, no real right or wrong answer, but something to consider kind of as we're coming into breeding season, especially for a lot of the seasonal guys. And then finally, we have something that's a little more new, which is selection intensity on the dam side. So this is something we maybe considered 10 years ago, probably more than 10 years ago, 15 years ago, as far as culling decisions. But it was really something that we could only leverage once we got viable sex semen. So basically, there are a couple of management things that influence this, and that major one is the choice to use conventional, the choice to use sexed and beef, or potentially using embryo transfers. And realistically, the reason why sexed and beef is so much better than conventional is it takes the randomness out of it. Now with sexed and beef, you can breed from your best dams and get replacements from them. And those dams that you don't want replacements from, you can breed them to beef, don't have to get a replacement. However, you can keep them in the herd because they're probably still very productive cows. And then finally, kind of one of those things that doesn't get talked about a lot, but by reducing cull rate, abortion rate, and DOA rate, not only are you saving on not having dead calves and not having to create more replacements, but you're actually being able to leverage selection intensity more heavily because you need less replacements. So that means you don't have to reach down for your 51st. You can stop at the 50th best. Clear as mud. Sweet. The next topic that we'll talk about today is accuracy. And as you can see, accuracy is still in the numerator of that equation. So the more accurate we are, the faster genetic gain we can make. And basically what accuracy is kind of the mistakes that we make when predicting an animal's genes. Pardon me. Excuse me. So there's a couple of ways that we can predict an animal's genotype. And the first of those would be pedigree evaluation. So that's where you look in your dairy comp, you find an animal's sire and an animal's granddam, or a grandsire, excuse me, and then you can take their genetic quality or their genetic merit, and then average them together and predict how good their offspring is going to be. The one caveat with this, and it's more of an issue than I had ever imagined, is quality of records. So in my years doing the genetic consulting deal, I worked with a handful of herds that did genomic testing and started genomic testing while I was working with them. And there was often times on those herds where we would find 40% mis-IDs for their replacement calves. So that basically means that somewhere along the line, whether the breeder wrote it down wrong, the breeder copied it into the computer wrong, or something got mixed up in the freshening pen, the sire that Dairy Comp said this calf had was incorrect. So 40% is probably on the high side, but I haven't seen anything that was much less than 15%. So the quality of records is really going to influence how valuable your pedigree evaluations can be. The good news about pedigree evaluations is they're typically free. So it's an easy way to draw those lines between the dams in your herd without having to spend extra money. The next method of evaluating is daughter proofs. So that's where we take a sire, breed him to a bunch of cows, and we look at his daughters. How well did they perform for milk production, DPR? How well did they breed back with? All these different things. And then we figure after we've got $200 of information, we can say with about 80% accuracy how good that cow is, depending on which trait we're looking at, right? And then finally, the last way is genomic testing. And and that's 70 to 75% accurate. And the way that happens is basically we'll go through the genome, look for different SNPs that we know mean something, and then compare the SNPs with other similar SNPs of populations, or compare that with other populations that have similar SNPs, and then look at how they did phenotypically, and make a guess on the genomics or the genetics there. So, with that being said, it looks like daughter proofs are the most accurate. Why are we screwing around with the genomics thing in the end if daughter proofs is the best way to go? The important thing here is you can't look at just one piece of this genetic equation and focus all your weight on it. A lot of these, especially these 4 traits here, interplay with each other fairly heavily. So if we look at generation interval, that's kind of to be where we look next. And the average age of your parents is basically how we define generation interval. And really, it's answering the question of how fast are those new genes being replicated, and how fast are they creating the next generation, I guess. For example, an elephant's generation interval is typically 25 years, and a fruit fly's generation interval is closer to 10 days. And that is the reason why geneticists in labs use fruit flies rather than elephants to do genetic experimentation on, right? Because they can see that genetic gain within their lifetime, or realistically within a couple months rather than 3 or 4 lifetimes. And the other thing that's important to note with this, I guess, is how it ties into accuracy. And the issue with daughter proving is that it takes a long time. You have to have your bull born. The semen has to be collected by year 2. You get that into a cow and it produces progeny in year 3. That progeny then has to grow up, get bred, and freshen in. And that takes about 5 years from start to finish. And then we have a very good estimate of how good that sire is, but it took us 5 years to get there. Whereas as on the first day that that calf is born, we can take a DNA sample, send it off to the lab, we get the genomics back, we know exactly how good that bull is going to be, and we can start collecting semen and using them in our herds right away. No delay. So because genomic test takes about half as long, it's more advantageous to sacrifice that small amount of accuracy to essentially double your generation interval, or I guess half your generation interval. And then finally, we look at genetic variation. And genetic variation is kind of the simplest and the most complex all at once. And essentially what it is the presence of gene sequences within a population, and really the frequency of those gene sequences. If there's none of them, we can't breed to get there. And that's especially important when we're considering multiple traits. So I don't know how many of you have looked and tried to find a Holstein with negative stature, positive DPR, I don't know, polled, and A2A2. Once you start looking at 4 different traits and you need to find all of them, it can be difficult to find all of those within the population of one breed, I guess. So that's why I think that restricting your population to one breed can kind of have a negative effect on your overall breeding plan in a lot of cases.
I have a question from the chat, Lane.
You bet.
Do you know if daughter proofs are helping to improve genomics over time and increase the accuracy of genomics?
Yep. Yeah, so that's a really good question. That's definitely, daughter proofs are basically what genomics is based on indirectly. Genomics, let me figure out the best way to put this. So genomics is basically taking a snapshot of the genome of a cow, and then it compares it with other similar cows with the similar genome. And then taking that, I'm not explaining this well at all. So once that's all kind of figured out, I guess, it compares the phenotype of those cows with similar genes, and that's how the prediction is made. So yes, more daughters and more data, that all feeds into the same database, but it's not necessarily influencing the genes or the genomics directly. Where it does play more of a factor is as we get more accurate by adding daughter proofs, because the CDCB doesn't stop counting at 200 daughters. It'll go 2,000, 4,000, infinitely, essentially. As much data as they have on a bull's daughters, they will collect. And then they can look back and see, okay, now that we have 2,000 daughters on this bull, we have a very precise idea of how good that bull is. This is what his genomics said. How do we make the two line up, and what kind of math needs to happen to get the two numbers to be as accurate as possible, right? I guess, was that a good explanation, or an acceptable one, I guess?
It makes sense to me. As your generations and sample size increase over time, that would be like the essentially like phenotype of the genotype that's being expressed. You'll have more proof for that as time goes on.
Exactly. So really, it's just a massive data compilation, and that helps drive all of that.
Awesome. Thank you.
You bet. Thank you. Okay. And now looking at this last factor here, which is inbreeding depression. So basically, what inbreeding depression is the reduced in predicted performance based on how related the parents are. So basically, when you have genomes that are very similar, it increases the likelihood that you're going to have what are called haplotypes or recessives. We know a lot of those haplotypes and recessives. A lot of mating programs try and keep them from happening, but they're oftentimes still kind of, there's a lot of them that we don't know about yet, and are affecting the offspring even without our knowledge. So by having more uniform populations that we're breeding from, it does increase our inbreeding. However, similar to how accuracy and genetic interval kind of play together, selection intensity and inbreeding depression almost have a similar thing. And the reason why inbreeding, as you can see, is kind of increasing, especially since the output of genomics, is we all want cows that breed back easier and make more milk with less inputs, right? That's all what we're looking for. So we're all breeding for a similar kind of cow. That means there's going to be genes that all of us are favoring, and it's going to create closer and closer, more inbred populations. Is that necessarily a bad thing? Probably not, in my opinion, right now, today. Because we're making more selection intensity. We're getting more genetic gain by creating a more uniform animal that makes more milk. However, they happen to be more related, and that's this inbreeding depression is slowly getting to be a bigger and bigger step, and the selection intensity is getting to be a smaller and smaller step. So eventually there will be a point where inbreeding depression overcomes that, and actually it will have a negative effect. We haven't seen it yet today. To hang tight, that is very possibly going to change over the next 10 years. But yeah, that's my spiel on inbreeding, I guess. If we want to get into more of a discussion on that, I'm happy to. And then that's more or less an overview of how that genetic progress equation interplays, and how genetic gain is actually gotten. I hope that kind of helps you guys understand why maybe you're doing some of the practices that you already are on your farm, or why maybe you should consider some of these other practices that maybe your neighbors are doing but you haven't gotten on board with. Just understanding the mechanics of that definitely helps me make decisions that feel more informed. Questions on that? And then just kind of another helpful place to look. I know talking with Jessica earlier, we discussed walking through a bunch of the individual traits that come on your proof, or whether you're looking through the bull book or catalog or online. seeing what those traits like DPR, milk, pounds, PL all mean. But honestly, that gets to be a very dry discussion. And it's much easier to just kind of use this link as a resource. So what this link will do, I wonder if I can open it. So what this link does is gives you kind of a list of all the different traits within the CDCB catalog. So it talks about what all these traits mean, as well as how they're evaluated. So it's a very useful handbook, I guess, if you're thinking about maybe changing some things in your genetic strategy. That is more, you guys can't see that. I'm sorry. I was looking at the link and I thought I was showing you guys stuff, but I wasn't. So either way, go on and click that link. I don't know if we can throw it in the chat so everyone else can get to it.
Yeah, just put it in the chat and I can just share it so people can take a quick look at it.
Sounds good. I don't have anything specific that we need to look at for today, but it's just really a good resource for figuring out what different traits mean, I guess, so that you can create your own plan. And I'm realizing that did not take an hour, but that's what I had for you guys today. So I apologize that I didn't use all of my time.
That's also okay. We're here for quality over quantity.
That's what I was hoping you'd say.
So that website looks like a really good resource. I had a question about sexed semen. Is that so? I'm sure it's very complicated. I don't even know that I would understand all the parts behind that. But is it a pretty typical, like, humans, males can give an X or a Y, females can give an X or an X? Is that also the same, or does something sort of like have the potential to change with cows in the canal?
Nope. So it's exactly the same. Males have an X and a Y, the females have an X and an X. And basically, how sex semen works is by leveraging that fact itself. So what's kind of cool is the X chromosome weighs 6% more than the Y chromosome, and that's how we sort everything. So there's essentially two methods of how that's done. One of them uses a centrifuge, and so the lighter male sperms spins to the outside, and the heavier female sperm stays to the inside. Otherwise, the method that Geno uses and ABS uses actually involves, like, it's almost like a Star Wars episode where they dye all of the sperm with a luminescent dye, and then they run them through a glass chip, single file, and all of them are oriented like this, because a sperm cell isn't a round deal, it's almost like a plate. And they orient them all in the same direction running through there. And there's one laser that detects the luminescence. And if it's 6% darker than a female cell, it goes down and the next laser kills it. And if it's the same luminosity as a female cell, it'll just keep on following through right into the vat. So our product actually has if you put it under a microscope, you'd see a whole bunch of living swimmers and a bunch of them with their tails shot off. And the ones with their tails shot off are the male cells. Yeah, it's pretty cool.
That is pretty cool.
I do have a question. So we know that a lot of our cohort uses balls still. So what factors do you think they should consider when choosing a ball for a grazing system, and how frequently they should introduce a new ball or switch the ball to introduce new genetics and to avoid the inbreeding on their farm?
Yeah, so using herd bulls is not my forte. I'll say that right off the bat. But I guess if I was going to give you some tips, I would say buy a purebred. Don't buy your neighbor's crossbred bull, because that's going to give you a lot of variation in subsequent generations. And that's just because of how those sperm cells are created. It's going to take a random half of the sire's genetic material. And it doesn't matter if it's a Holstein or a Jersey, it's going to take a random half. We don't know what that half is going to contain. And every cell is going to have a different ratio of Jersey or Holstein genetics carried on it. The second thing would be, if you're going to go out and buy a herd bull, ask for a genomic test. You can take the genomic test and send them into the CDCB. They'll be able to give you a better idea of what that genetic value is. And you can compare that then to what's available on the AI. You could compare it between different bulls out there. It's something that's done fairly frequently within the beef industry already, I know. I don't know how well it is, or how well accepted that is in the dairy side, just because there's less guys who use herd bulls within the dairy industry. So genomic testing is a very good option to know that you're getting a good quality bull. And as far as avoiding inbreeding, it kind of depends on your system. I'm sure you could go through and document who's sired by who still, but realistically, if it were me, I would be changing bulls at least every 2 years anyway, just because by the time a bull's 4 years old, it's probably fairly aggressive and ready to go anyway. So a lot of the guys who I know who use herd bulls don't keep them much more than 2 years old anyway. Any other
Awesome,
questions?
thank you. I do have another one. So let's say that there's some farmers out there that want to transition their cows from confinement to grazing systems. Considering their genetic potential, what do you think they should focus on?
So that's a difficult thing to answer without knowing what that confinement system was already like, what those cows already started with, right? I would say feet and legs and realistically stature and total body weight, I think, are some things that are underrated. Just the more weight that you're putting on those four hooves, we don't have any traits that breed for wider hooves, so you can't make the pressure for each foot less any other way than taking total weight off the animal. So I think that is a big thing. Plus, they can tend to be more efficient because we figure there's about 2% of live body weight is needed 2% of the body weight is needed in dry matter just to maintain that body weight.
Awesome, thank you. No more questions on my part.
Anything from the group? I see we're all the way up to 7 in attendance.
I just got a compliment. Thank you for being informative. It was math, but it was easily broken down and can be used to extrapolate good habits to keep track of. I agree.
Awesome, because that's exactly what I was aiming for. That way, when someone pulls up your driveway, you can tell if they're actually selling you something or just blowing smoke.
And then I have another question from Kathy. We used quite a bit of Scandinavian red breeding about 10 to 12 years ago, and then did use one of our own bulls that was 50% Scandinavian red on our heifers for 6 months plus. It seemed at that time there was higher percentage of somewhat sickle eggs on more of the Scandinavian red animals than our Holsteins. Is that a trait that has improved?
I would say yes within the Norwegian red population for sure. I haven't paid nearly as much attention to our counterparts with Viking genetics as I had in years past. But within the Norwegian red breed, feet and legs has definitely improved drastically from what it was, as well as udder composite is significantly better.
Awesome. Anybody have any other questions they want to type in before we wrap up? Let's give 1 minute so people don't feel rushed typing. I hate feeling rushed.
Absolutely.
When Kathy added, that was great because the udders needed improvement too.
Absolutely. Kathy, you hit the nail on the head there. We started with Scandinavian reds close to 10 years ago at the farm back home, and I saw some train wrecks out of it. I was a little bit nervous coming back and working with the product again, to be honest. But the other cool thing about the Norwegian system that kind of goes along with expensive vet prices is everything is documented extremely well. So like every hoof trim, every antibiotic, every vaccination, every breeding, and all the milk production data all goes back to the co-op. So Gino is able to keep extremely meticulous records, and they have actually genetic advisors from the co-op that design plans for individual farms. And basically by using that as leverage, we're able to have way more data and way more accurate data backing our proofs than a lot of the stuff in the US that is, well, I've worked with a lot of dairy comps, and going back to the idea of potentially having 40% incorrect sires in the proof, that all went back to CDCB, and they counted the milk production data and all that incorrectly for all those sires as well still.
Wow.
Yeah. Not to say that it's inaccurate, and those inaccuracies are kind of accounted for. It's still the best system. It's not perfect.
All right. And yeah, Kathy said she might have to try the Norwegian red again.
Well, you got my email.
All right, well, if we don't have any more questions, we can get wrapped up here. Thank you again, Lane, for that. That was really, really digestible and a lot of data. I have one more, I'm sorry. Would it be beneficial to use genomic testing and other mating methods in addition to using bulls raised in grazing system?
I'm not sure that I quite understand that question. Sorry.
That's okay. Beneficial to use genomic testing. I guess he's asking if there's like an eclectic approach to maybe having grazing bulls and then also using genomic testing for, I guess AI would be, I think to me there's only two mating methods, AI and a bull. So
Okay. So I would
would it be
say
beneficial to blend them?
if we're talking about genomics on the bull side, yes, it's worth it as long. Well, genomics in any case, it's only worth it if you're going to use the data. So you could genomic test all of your cows in your herd, but if you're going to breed them with a herd bull anyway, what is that going to actually? What effect is it going to have? None, because you're not making a decision as to who gets to keep a calf or make a replacement and who gets bred to beef. If you're using that to differentiate between animals, then genomics can be a very viable option. The one other caveat that I need to throw out here while we're on the topic of genomics, don't buy a genomic test for your crossbreds because it doesn't work, no matter what anyone tells you. It's not accurate.
Okay, thank you. All right. Well, thank you again. Thanks everybody for being here and for interacting. Thanks for trying out this new platform with us. This is our first time. Jess, Lane, and I have to stay on to make sure everything gets